shared, and I also have uploaded this paper. So the idea would be that, and there would be one part last that I couldn't finish, and just one more lecture, which I'll indicate the chapter, and it will be like a small part, and maybe one question from that, and the rest of the questions will be from only the materials that I have shared, the slides I have shared, and one part would be what I have what we'll try to do is 75 to 80% will be on the topics that we went over, and the 20% will be on a paper that I'm just going to talk in brief about now. And the idea would be that so this paper is very seminal, and it's relatively recent, and it came out in 2011, and I just wanted to give an example, which is a recent paper. It uses most of the recent techniques so that you guys get to know about them, and it also kind of have changed the way we think about a certain kind of field of neuroscience. And if you have interest, you can follow up their work in the last since the paper was published in the last 9 or so years, and how they have kind of started with the field and kind of changed the way we think about it completely, right? And so that's why, and this is their first paper when they started off in this field, and that's why I wanted to go over it. And the questions would be more about your understanding, not the technicalities or the technical details from this paper, but rather what we'll focus on is your understanding and what are the kind of the major points they wanted to get through in this work, and how this is novel, and try to understand why is it so important. And we'll do 3 or 4 questions from this paper, and the rest of it will be very textbook, what you went over. And the only reading material for those questions you would need is the PDF of the textbook that I have shared. Yeah, so this paper, and as I said, I'll go over it very quickly. I won't go through the details of it again. You guys can read the paper, and so the file that I've uploaded has all the details that you need, all the supplementary data. I think, yeah, everything is there. So the idea here is that for many years, for even 100 years or so, we knew that our skin has receptors. The sensory neurons innervate our skin, and it innervates not just the skin, but also the hair follicles. And that's how we sense somatosensory stimuli, which means that so the skin is the largest organ of our body, and it's exposed to all kind of stimuli around us. And if we don't perceive them properly, we cannot function, we cannot go about our daily life. And it can go from, you get up in the morning, you drink tea or coffee, doesn't matter. You want to see it's hot enough or not when you're making the tea, or you want to have a hot cup of tea or coffee. So you need your somatosensory system for that, right? So you might have a pet and you want to pet that, a dog or a cat, and that you need your pet needs the somatosensory system to understand your affection. Or you also need to make sure that you kind of share the warmth with your pet. And things like that. So just like your visual system and auditory system, somatosensory system consists of a very integral part of your daily life. And this paper basically kind of gets to the very cells and the types of cells that innervates our skin, and kind of try to break it down to kind of its components, and try to understand how each of this component, the sensory neurons, work. And why it's so similar, and why is it so important? And that is kind of what the point I'm trying to get across, that try to understand this was lingering questions for many years, and why it has to be solved, right? So say you have robots now, and you want a robot with kind of a somatosensory system with acuity and the sensitivity of humans, so it can kind of do the tasks that you have given them, and kind of very naturally do it. But if you can design a robot like that without understanding the somatosensory system that exists in our normal humans. So having said that, so they just to look at the abstract, and I would also like you to understand how to kind of try to learn how to read a paper. And so each paper, so the difference between a paper and a textbook is that a paper, it's a research project that has been undertaken by a scientist. It's sort of an opinion with evidence, which what I mean by that in the sense that the scientists started with a question, they formed hypothesis, they tested the hypothesis, and they provided evidence for it. And it's up to you and up to the peers and up to the experts to actually kind of try to make a sense of it and kind of give a judgment, okay, we believe it or we don't believe it. And it's up to the scientists that will, for years, they will come up with more evidence and they will build on it. And that's why, so what I said was, if you have time later, go to their work and see that how they have kind of provided evidence and built it up on this paper. And 1 second. Yeah, so built on this work, and then they kind of solidified, okay, like what they had found was true. And there are other scientists that made counter arguments and say, okay, like, okay, this part is true and this is not. So, and what is the difference between that and a textbook is like, when this last 10 years of work, eventually kind of scientists agree on it, and then a part of it will become from a textbook. So the point here is when they started something that novel and that provocative, the point is not that it doesn't become a fact. They just provide the evidence, and it's up to the reader to judge on the evidence, up to the scientist or you that to read the paper and say, okay, I buy this and I don't buy this. And it's kind of an argument or opinion with evidence, as I said. So we try to kind of read this paper. We'll just try to go through the paper in a very short. And I said that what they're trying to define is the innocuous touch receptors in the skin. So the innocuous touch of the skin is detected by distinct population of neurons, and which are the low threshold mechanoreceptors. So there are three kinds of mechanoreceptors: A beta, A delta, and C. And if you remember the myelination patterns of sensory neurons, so the thickest, the more myelin there, A delta, less is A beta, then A delta, and then C. No myelination here. So the fastest, the kind of medium speed, and the slowest communicating neurons. And so these are only, and what I mean by low threshold means that which are not detecting high threshold stimuli. It's like not detecting the pinch is not for the pinch neurons, but if you kind of take a brush, like a pen brush, and gently, kind of sweep over your skin. So the neurons that are detecting that are these low threshold mechanoreceptors, and they have three types. And so what they did was they kind of visualized how these neurons innervating the skin. So they're studying it in mouse. And the advantage of mouse being that a lot of genetic tools are available. So what it helps us doing is you can kind of visualize each of these neurons, each of kinds, and then you can manipulate it. You can excite them artificially, you can silence them, and you can ask, okay, what is the consequence of exciting or silencing them on animal behavior or neuronal physiology? And so there are three kinds of hair, basically. We also have it in different proportions. It's called all, Austrian, and zigzag. So these are if you look at a hair under a microscope, it will actually look pretty different depending on the pigmentation and how smooth or rough they are. And they are innervated by a unique and variant combination of LTMR. So each skin is pretty differently innervated, and so that the hair are kind of distributed all over a body at different proportions, depending on what kind of sensitivity that part of the skin requires to transmit. And also each distinct mechanoreceptors, and also these neurons are going into the spinal cord and different regions, so that they're innervating again a different set of neurons which are transmitting the information to the brain. So they kind of looked into that too. And so I won't go to the text, which you can you guys can read about. And so these are basically what I want to show you how things look like under the microscope. So, modern neuroscience or modern biology uses a lot of fluorescence. So what I mean by that is that you can, using fluorescence, because when you look at a black and white picture, you just got black and white. You can observe multiple components together. What fluorescence helps us with is, since fluorescence come at different flavors, that is different wavelengths, they can emit and they can absorb and emit light at different wavelengths. So we can kind of visualize multiple components at the same time. So it can be a red channel, far red channel, green channel, yellow channel. So we can look at 4 gene expressions at the same time, 5 gene expressions at the same time, depending on the sensitivity or what kind of microscope you have at your disposal, right? So what they did was they found that the LTMRs so they're looking at the CLTMRs, or the slowest conducting ones. And what they see is that they express different kind of proteins, which you can by yourself go at the details of it. And they show that the CLTMRs are expressing this protein called TH, or tyrosine hydroxylase. So in this, we are just using these genes as markers for the neurons we're looking at, not necessarily studying the role of the genes itself. And what they did was they kind of recorded from these neurons, and they found that when you increase when you give different kind of force, our neurons are kind of tuned to different kind of forces and not thermal temperature. So these are mechanically sensitive. And then they look at their anatomical, what kind of ending. So this is a hairy skin. So these are the hair follicles. If you look at the hair from the top, this is one neuron is innervating at this area. And then so this is called arborization, which you will find in a lot of details in the text. And what it does is each neuron is innervating a particular part of the skin. And what they're labeling here is kind of patchy, kind of random neurons they're labeling. If you remember the first class, the Cahill's experiment. So this looks like one neuron can innervate in this part of the area. If you zoom in, they look like this. So the hair will go in kind of in middle of it. And this is how the one innervation looks like. And this is in fluorescence, exactly the same structure. You can see that these are kind of, if this is a hair follicle standing here, then innervation is all around it. So if the hair is deflecting, it's kind of sensing it. So if you really think like a gentle breeze blows on your skin and your hair deflects, and how do we detect it? You would think something like this is taking that into account. And then, so they found another kind of, now it becomes a deltas. It has a different identity, but they look very similar. So they look very similar. They are kind of innervating part of the skin in combinations. They have different thresholds, and the idea is that they have different markers. So once you have different markers, transgenic animals that can label the neurons like this, only this and not the other one, and then you can excite them or silence them and eventually test what would be the difference in behavior. You can also record from them and say, okay, this only tuned to only this kind of stimuli and not anything else. And same thing goes with another type, which will be now your A betas, or the first. And you can see this beautiful structure. And if you remember, all neurons come with different flavors of glia, and they're innervating it. And in the following papers, they go on try to test what are the functions of this glia. Then, as I said, the fluorescence and this genetic tools, what allows us to do is looking at different type of neurons in combination now. So they have now mixed one type with another type and see same hair is innervated by 2 kinds. And in later papers, they come up with they ask like, okay, so if this one neuron is, you'll see only 2 sides, see here 2 sides. So this hair, when it's deflecting this way, it can sense, not this way. So now it's kind of a direction selectivity. So your hair, if you could, like you take a huge hair and then you deflect to different directions, they will only excite one kind of neurons and not other. That's how we kind of, our hair has this direction selectivity. So if you kind of touch your skin from one direction to the other, one set of neurons will be firing. And if you go for the other direction, another set of neurons would be firing. And if you go for any direction, then another set of neurons would be firing. Yeah, and then as I said that, eventually they will go to the central nervous system, the spinal cord, it will go to different depths and it will form these columns. And there will be neurons here that are sensing only this information from these neurons at different combinations and then compute them, send it up to the brain. And that's how, so it's not a the central nervous system or in living organisms is a very distributed system. So there are computation at different levels. It's not just uh, it's not just one, this one center that is uh, processing everything. So and then what they did was they injected dye, at the periphery, and which will come back, and then they show that these neurons are, going to one part of the skin is forming kind of one innervation. So you have to go through these details, and this will be a kind of assignment and test that what we understand from this paper. And as I said, like we won't get into the very technical details, won't questions about that, rather about the very kind of background of this paper and the very basis on which they have built this work on. And you will see a lot of biologists, especially molecular biologists and geneticists, like I would be one of them, and we would always say, a story, and you won't hear this in any other kind of science. It's kind of like why a science should be a story. But biological systems are kind of a very complicated, and we can't just ask one question. So if you just ask, okay, what are the neurons, and you publish a paper and it doesn't really make sense, you kind of draw a complete picture, or kind of complete picture, to be really make it convincing. So I know you often say, like, you basically in our papers, what we do is we show really good papers. We show just a same apple from different sides to convince people, okay, it's an apple, is not half of an apple, it's not a quarter of an apple, it's not a picture of an apple, it's not a 3D printed apple, it's a real apple. So that's why, since biological systems are so complicated and kind of, so we often tell them like a story that kind of build a narrative around it. And so the idea would be to kind of get a flavor of that and where this work is coming from and where is it taking us, right? So that would be that, and I have shared this paper, and yeah, and then if you have any questions, you can ask. Yeah, anybody? Okay, there are no questions. So then we'll just, I'll send you more materials for the test, and if you have any questions, you can ask on the Teams, right? Okay, bye.